Bismillahirrahmanirrahim. Today I am going to discuss sensory receptors. Sensory receptor. In a sensory system, a sensory receptor is a sensory nerve that responds to a stimulus in the internal or external environment of an organism. In response to stimuli, the sensory receptor initiates sensory transduction by creating action potential in the same cell or in an adjacent one. The sensory nerves convert a stimulus to action potential through receptors. The whole process of converting the stimulus to action potential is known as sensory transduction. The sensory receptor may be a specialized portion of plasma membrane or a separate cell associated with a sensory nerve ending. This is a sensory nerve which detects any change in the internal or external environment and then transmit this information to the central nervous system. Transduction. In physiology, transduction is the conversion of stimulus from one form to another. Transduction in the nervous system typically refers to a synaptic event wherein an electrical signal known as action potential is converted into chemical one via the release of neurotransmitter. In this diagram, the stimulus is light energy, electromagnetic energy which is emitting from the bulb. Reception is the eye, and receptor in eye detect and response to light. Transaction occur here. Light is converted from electromagnetic energy to action potential or a neuronal impulse. Transmission occur through sensory nerve, and these information are sent to the brain via the optic nerve. In the brain, these impulses are sent to the primary visual cortex, where light energy is transduced into image of the light emitting source. Transduction is the process of turning environmental information into neural impulses. In the eye, light wave create neural impulses. In the nose, chemical reaction from gases molecule create neural impulses. On the tongue, chemical reaction create neural impulses. In the ear, sound wave create neural impulses. Within the skin, pressure, pain, and temperature create neural impulses. Modality. A stimulus modality or sensory modality is a type of physical phenomena that can be sensed. Examples are temperature, taste, sound, and pressure. Modality means that we can sense these things but cannot see. Like temperature, we can't see temperature, taste, sound, and pressure, but we can sense them. So this is called modality. Here, the cold, the touch, the heat, and the pressure. In the skin, they are sensed, but they are not visible. Senses are vision, which is from the light, smell from the gases molecule in the air, taste is a chemical which is tasted by the tongue, hearing is sensed by hair cell in the internal ear, and touch on the surface of skin. The type of sensory receptor activated by a stimulus play the primary role in coding the stimulus modality. Type of receptor. Chemoreceptor responds to chemical stimuli like the receptor which are present in the aorta, and chemoreceptor responds to chemical stimuli. Mechanoreceptor responds to mechanical stress or mechanical strain, moment, pressure, and tension. Thermoreceptor responds to temperature, either heat, cold, or both. These receptors are present in the skin. Photoreceptor responds to light present in the eye. Baroreceptor responds to pressure. Osmoreceptor responds to osmolarity of fluids such as in the hypothalamus. Proprioception provides the sense of position. Nociceptor responds to nauseous or potentially nauseous stimuli. Hydroreceptor responds to change in humidity. So chemoreceptors are present in the aorta and carotid arteries, and they sense the change in the pH of blood or change in the carbon dioxide partial pressure. Classification by location. Sensory receptor can be classified by location. Cutaneous receptors are sensory receptor found in the dermis of skin. These are the various type of sensory receptor which are found in the skin. Muscle spindle contain mechanoreceptor. They detect stretch in the muscle. This is a muscle spindle which is found in the muscle belly and detect any stretch or change in the length or tension in the muscle belly. Golgi tendon organ are proprioceptors that are located in the tendon adjacent to the myotendinous junction. This is myotendinous means this is tendon and this is muscle. At this junction, this receptor is present and they detect proprioception. Classification by morphology. Sensory receptors near the surface of the skin are divided into two groups. Free nerve endings characterize nociceptors and thermoceptors. Terminal branches of the nerve are unmyelinated and spread throughout the dermis and epidermis. These are the pre-nerve ending are unmyelinated nerves and they are spread in the dermis and epidermis. This is the epidermis and this is the dermis, and these pre-nerve endings are which are unmyelinated are spread in this area. Encapsulated receptor comprise of the remaining type of cutaneous receptor. The rest of the receptors which are present in the skin are capsulated. Mechanoreceptor. A mechanoreceptor is a sensory receptor that responds to mechanical pressure or distortion. There are four main types in the glabrous skin or of human. Hairy skin. Glabrous skin or hairy skin of human. Pacinian corpuscles. They are Pacinian corpuscle, Meissner corpuscles, Merkel disc, and Ruppini corpuscle. Hair cell in the cochlea are the most sensitive mechanoreceptor in transducing air pressure wave into sound. These are the various receptor. Tactile Meissner corpuscles response to light touch. This is the tactile Merkel corpuscle response to touch. Pre-nerve ending which is nociceptors response to heat and pain. Laminated Pacinian corpuscles, they response to deep pressure. Ruppini corpuscles, they response to warm. So I will discuss these one by one. Pacinian corpuscles. Pacinian corpuscles are one of the four major types of mechanoreceptor response for sensitivity to deep pressure, touch, and high frequency vibrations. The structure of the Pacinian corpuscles is ovoid shaped and approximately 1 millimeter in length. The length is 1 millimeter. The entire corpuscle is walled by a layer of connective tissue. This is the layer of connective tissue which walled the corpuscles. It has 20 to 60 concentric laminae composed of fibrous connective tissue and fibroblasts separated by gelatinous material. These are the lamina. They are separated by gelatinous material from each other and they are 20 to 60 in number. The lamina are very thin, flat, modified Schwann cells. In the center of the corpuscle is the inner bulb. Here this is the center of the corpuscle and we have a bulb which is filled with fluid with a single apparent unmyelinated neuron ending. Pacinian corpuscle is innervated by myelinated nerve, but in the center of the bulb the nerve is unmyelinated. The Pacinian corpuscle is like onion. These are the lamina. They are similar to the onion corpuscle. In the center is a bulb, and in bulb is an unmyelinated nerve ending. Pacinian corpuscle responds to deep pressure. When pressure is applied, these lamina deform, and this deformity send impulses to the unmyelinated nerve, which is located in the bulb surrounding by the fluid, and the pressure is then converted into action potential. When action potential is generated, it is transmitted to the central nervous system. Location. These corpuscles are found in mesenteries, especially pancreas, and are often found near joints. They are found in deep subcutaneous tissue and are considered rapidly adapting receptor, which means they will not fire action potential throughout the duration of the stimulus, but rather will fire briefly at its beginning and end. When pressure is applied, they are stimulated and then they adapt rapidly. Functions. Pacinian corpuscle detect gross pressure change and vibration. These corpuscles are especially susceptible to vibration, which they can sense even centimeter away. Pacinian corpuscle cause action potential when the skin is rapidly indented, but not when the pressure is steady, due to the layer of connective tissue that cover the nerve ending. It is thought that they respond to high velocity change in the joint position. Pacinian corpuscle have a large receptive field on the skin surface with an especially sensitive center. They only sense stimuli that occur within this field. The Pacinian corpuscle was named after its discoverer by Italian anatomist Filippo Pacini. Meissner corpuscles or tactile corpuscle are a type of mechanoreceptor response or sensitive to light touch. Location. They are distributed throughout the skin but concentrated in areas especially sensitive to light touch, such as the fingertip, palm, sole, lip, tongue, face, and skin of the male and female genitalia. They are primarily located just beneath the epidermis within the dermal papillae. Structure. Missile corpuscles are encapsulated unmyelinated nerve ending which consist of latent supportive cell arranged as horizontal lamina surrounded by a connective tissue capsule. The capsule is between 30 to 140 micrometer in length and 40 to 60 micrometer in diameter. Here, these lamina, the swan cell, they are located transversely, and the nerve enter and pass through these lamina. This is the length and diameter of the corpuscles. A single nerve fiber meanders between the lamina and throughout the corpuscles. Location. They are distributed throughout the skin but concentrated in areas especially sensitive to light touch, such as fingertip, palm, sole, lip, tongue, face, and skin of the male and female genitals. They are primarily located just beneath the epidermis within the dermal papillae. Functions. Any physical deformity in the capsule will cause an action potential in the nerve. Since they are rapidly adapting or phasic, the action potential generated quickly decrease and eventually cease. If the stimulus is removed, corpuscle regain its shape, and while doing so, that is, while physically reforming, cause another volley of action potential to be generated. This is the reason one stop peeling one's clothing. When we wear clothes, at first we feel the weight of the cloth, but when these receptors are adapted, then we are unaware or we don't feel the weight of the cloth. Because of their superficial location in the dermis, these corpuscles are particularly sensitive to touch and vibration. But for the same reason, they are limited in their detection because they can only signal that something is touched the skin. Master corpuscles were discovered by the anatomist George Meissner in 1829 to 1905. Merkel nerve ending are mechanoreceptors found in skin and mucosa of vertebrates that provide touch information to the brain. These are the receptor Merkel nerve ending which provide touch information to the brain. Each ending consists of a Merkel cell in close opposition with an enlarged nerve ending. This is the Merkel cell and it is closely located with this cell. This is referred to as a Merkel cell neurites complex or a Merkel disc receptor. It is also called a Merkel disc receptor. A single apparent nerve fiber branch to innervate up to 90 such endings. One nerve can innervate up to 90 such receptors. These are the Merkel discs or cytoplasmic projection from this Merkel cell, and these are the granules found in the Merkel cell and desmosome found which bring these vesicle surface of the cell, releasing of neurotransmitter. They are classified as slowly adapting type 1 mechanoreceptor. Location in mammals. Merkel nerve endings have a wide distribution. Merkel nerve endings are found in the basal layer of glabrous and hairy skin, in hair follicle, and in oral and anal mucosa. In human, Merkel cell along with Meissner capsule occur in superficial skin layer and are found clustered between the ridge of the fingertip that make up fingerprint. Their rigid structure and the fact that they are not encapsulated cause them to have a sustained response in the form of action potential or spike to mechanical deflection of the tissue. They are the most sensitive of the four main types of mechanoreceptor to vibration at low frequency around 5 to 15 Hz. Because of their sustained response to pressure, Merkel nerve endings are classified as slow adapting. Merkel nerve endings are extremely sensitive to tissue displacement and may respond to displacement of less than 1 micrometer. Ruppinian ending, named after Anglo-Ruppini. The Ruppini ending is a class of slow adapting mechanoreceptors throughout to exist only in the glabrous dermis and subcutaneous tissue of the human. This spindle-shaped receptor is sensitive to skin stretch and contribute to the kinesthetic sense of and control of finger position and movement, and their response to stretching of the skin. As the system is changed from long essay question to MCQs, I have decided to add some MCQs to my lecture, and in this lecture there are 2 MCQs. So the first MCQ is, what type of receptor cell is responsible for transduction pain stimuli? These are the 4 options. Please write the answer in comments. MCQ 2 is, what type of receptor cell is involved in the sensation of sound and balance? Write the answer in your comments on the lecture. So thank you. See you in the next lecture.